Hello, welcome to this biology video on diffusion. This video is actually diffusion part 2. So if you have not looked at part 1, please go back to that video and have a look at it because that covers some basic aspects of diffusion. It goes through the definition, what it is, and how it happens in the various factors that affects the rate of diffusion. So it's really worthwhile going through the part 1 of this video before you go into this, because this covers the specifics of biological systems in animals and plants and how these systems are adapted to increase the rate of diffusion. So it'd be really useful if you have not already watched part 1. Please go back and take some time to look through that video. So in this video we are going to be looking at how human systems, and this is common to most mammals, how the lungs are adapted particularly to maximize the rate of diffusion. Small intestine, how food after digestion moves into the bloodstream in the small intestine, and how small intestine is specifically adapted to maximize this process. Semi-permeable membranes, which is the cell membrane which is in every cell of human body or tissues, how the substances can go into, diffuse into and diffuse out of cells. We'll be looking at specific cases where some molecules are able to and some molecules are not able to, hence it's called semi-permeable. So we will be looking at that using a cell model. We'll also look at specific parts of plants such as leaves and roots, and how these parts are specifically adapted to maximize absorption of substances or taking the substances in and out of these organs, leaves and roots. For instance, carbon dioxide and oxygen has to go in and out of leaves, and how the leaves are adapted for that. Roots, minerals and water are taken in through the roots from the soil. So we can look at all of these systems, how they are adapted to maximize the rate of diffusion. To understand how diffusion works in small intestine, it's useful to look at a model, an experiment setup, which is normally done in schools, where you have a plastic container, a bag, which is semi-permeable, and the plastic container itself is porous, which means it's got tiny holes in them through which substances can move out or in. And such a tube is called a whisking tube. You might have seen this, and we're going to use that model this time around to model the small intestine. Now later on we can use the same model to describe and understand how diffusion works in cells. So we can use that model in two different ways, but this time around I'm going to use that to explain how the small intestine works and how it is really well adapted for that purpose. But first of all, we need to understand what happens in the small intestine. We're going to have a separate lesson on digestion, so we're not going to look into the details of the molecules that are involved, that's getting digested in the small intestine and which is absorbed or diffuses into the blood. So we'll not be looking at the details of how the digestion happens, but we're just interested in how the diffusion works in the small intestine. I hope it makes sense. So let's assume this part here represents our small intestine, and there is food inside here. And for food, to keep it simple, we're going to consider starch, and we're going to assume that digestion is taking place here with enzymes. Enzymes are chemicals which we're going to deal later in a digestion lesson, but for now all you need to know is enzymes actually break down bigger molecules of food to smaller molecules. Okay, so that's all for now on enzymes and on digestion. So the focus should be on diffusion, how the broken down smaller molecules move from inside the small intestine to the blood, and how it's circulated around the body. Okay, so to understand that, we need to look inside the small intestine. So I'm going to show you a diagram which shows the cut open. If you cut this intestine open, how does it look like in cross section? So you're going to look at a cross-sectional view of the small intestine. So let's have a look at that. See, that's how your cross-section of a small intestine looks like. Okay, so remember, this small intestine is modeled by this tube here. Okay, so this is like a cut open. If I cut this open, the small intestine has a cross-sectional view like this. It's a bit exaggerated. The view is it doesn't exactly look like that, not to scale, but it just is to illustrate a point here where you have something called villi on the walls of the small intestine. You know, these finger-like projections, they're called villi. If you look at just one of them, it's called villus. So that's the plural of villus. So you got thousands and thousands of these lined on the inside wall of the small intestine. The purpose is, so when the food passes through this this opening through the small intestine, it comes into contact with more surface area. So this maximizes surface area. So the millions of villi we have inside the small intestine, I hope it millions or if not thousands, that increases the surface area enormously, which means the diffusion happens at a faster rate through all the villi that's there. Okay, so how else it is adapted? One it is surface area, and the second one, if you look at each individual villi, so here is one made to look bigger. So what you have here is one of these villi. Okay, so how does that look like? You can see there, in the villi itself is very thin. Can you see that there? It's made of one cell thick wall. Okay, so the wall is made up of one cell layer. Okay, so single layer of cells make up the villi wall. Okay, so the wall is made of a single layer of cells. So that means it's very, very thin. So as you remember from your part 1, if you reduce the distance between the diffusing substance, the distance the substance has to diffuse, the rate becomes faster. Now the food is here. Okay, so the food is in this area. This is the outside of the small intestine. Okay, so food is digested here. So you got the small molecules in this area, and it has to go through that wall. So food is here, which is here. It has to go through that single wall there and straight into the bloodstream. And you have a very good supply of blood in each of these villi. As you can see here, that red line that shows the blood supply line. And because you have very good blood supply inside the villi, food has to just go through that single layer of cells straight into the bloodstream. So the rich supply of blood is another adaptation of small intestine. So two adaptations. One is the thin wall, which is made up of single layer of cells, and the second adaptation is the rich supply of blood. Okay, of blood. So second one. The third one, you can see, you can also describe the blood being constant motion, which means it's constantly moving through that way. So it comes in, takes the food supply and goes away. What that means is fresh blood keeps coming in, which means the difference in concentration between the digested food over here and the blood, the amount of digested food in the food molecules in the blood is maintained bigger. So it's always maintained bigger. So that means the diffusion rate is faster. So this is a bit hard to understand, but if you think about this, the higher the difference in the concentration of any substance that's diffusing, the faster will be the rate of diffusion. Now if you remember that from the part 1. Now as the blood keeps moving through these capillaries, very thin tubes in the villi, what happens is, as fresh blood arrives into the villi, you have a bigger difference in concentration all the time. So it helps that the blood keeps on moving in that way. Let's have a look at how lungs is adapted for faster diffusion or efficient diffusion of gases. Now, as you know, lungs exchange gases. What we breathe in comes through the trachea into the bronchus, and through the bronchioles it goes into this air sac called the alveolus. So we're going to be looking at how efficient, how it is adapted, both lungs and in particular how the gas exchange takes place between the alveolus and the blood. These tiny air sacs, there are around 300 millions of them. When I say air sacs, I mean alveolus. One of them is called alveolus, and if you're talking about all the 300 million of them, roughly, we can call them as alveoli in plural. Okay, and that's the lungs is both lungs are a special organs which are adapted for efficient exchange of gases. And as you know, lungs' main job is to take the oxygen into the bloodstream and to get rid of the waste carbon dioxide. Both happens by diffusion, and it's really important that we recognize that and remember that throughout this discussion. Okay, so let's have a look at how the bunch of alveolus or alveoli look like. As you can see, the bunch of alveoli, which is also referred to as sometimes air sacs. So it was one of them. Here's a bunch of them, and they are surrounded by this blood capillaries, very thin blood vessels, and the sheer number of the alveoli present in each of the lung, as you can imagine, 300 million of them, increases the surface area. So it's got very high surface area, or very large surface area, is due to the number of alveoli. So it's enough for you to say large surface area, and because of the number of alveoli present. Let's have a look at one single alveolus and how the gas exchange actually takes place, and the adaptations of the alveolus or the whole system for efficient exchange of gases, which in this case, as you can see, the orange arrow comes in, contains the incoming air. It's a mixture of air, but the oxygen is able to diffuse through into the bloodstream straight to the blood cells. As you know, the red blood cells are the ones which carry the oxygen. And so the alveolus is again made up of one single layer of cells. So it's very thin walls. The alveoli walls are very thin. In case it's one single layer thick, which means it's extremely thin. And you also have a thin coating of a fluid inside the alveolus, which means the oxygen can dissolve in that liquid, which makes it easier for it to then diffuse into the blood capillary. So the blood arrives into the capillary and moves away from the alveolus, and as it moves, the carbon dioxide, which is shown by the blue arrows here, move or diffuse from the capillary blood into the alveolus. Oxygen diffuses the other way around. The direction is because there is more oxygen concentration here compared to here, and as you know, the diffusion takes place from an area of higher concentration to an area of lower concentration. The same works for carbon dioxide. You have more carbon dioxide concentration in the blood compared to the alveolus, so it moves that way and it's breathed out through the lungs. Okay, now the blood also, so we know the thickness is the main factor. It's extremely thin. You've got two layers of cell for the oxygen to pass through. One is the alveolus, and then the blood capillary being very thin, again one cell layer thick. It's able to diffuse easily through the two layers of cells straight into the blood. Okay, so that's one important adaptation. The third one being the blood movement. Now the blood keeps on moving this way, which means the blood which contains the oxygen keeps moving. Fresh blood arrives, which has got lower concentration of oxygen and high concentration of carbon dioxide, which means the difference in the concentration of the oxygen between the alveolus and the blood is maintained higher. So there is a higher difference in concentration between the alveolus and the blood all the time because of this movement of blood, and that makes it even more efficient. Okay, so the movement of blood means it maintains a higher difference, which is larger difference in concentrations of oxygen and carbon dioxide in the blood to the alveolus. Okay, so just to summarize, very large surface area due to the millions of alveoli present in each lung, and you have the walls of the alveoli made of one single layer of cells, which means it's extremely thin, which makes the diffusion distance very small, so the diffusion is faster. You also have the blood moving, which maintains the concentration difference between the alveolus and the blood. So there's a higher difference in concentration of oxygen, which means the diffusion into the blood is faster. So here is a summary now of the adaptations of lungs, in particular the alveoli, for efficient diffusion of gases. So if you can read through that, that is a summary. All the three main points are there. Let's have a quick look at how oxygen diffuses from the blood vessel into tissues and what adaptations are there. Two main adaptations, one being the blood being mobile, moving through the blood capillary. So you need to think of that as a main factor. So the blood keeps moving that way through the capillary. Cells are around the blood capillary. These arrows indicate movement of a certain substance which goes from the blood into each of these cells. Okay, so you can imagine the cells closest to the capillary will get the maximum diffusion, and then the oxygen diffuses from that cell to the adjacent cells. Okay, so there will be a higher concentration of oxygen here than here, obviously, because it's closer to the blood capillary. So the blood capillary oxygen concentration will be the highest compared to the oxygen concentration in the cells, because the cells respire, they use up oxygen and produce carbon dioxide. Okay, so the concentration of oxygen will be lower here, so that makes sure that there is a concentration gradient, makes the diffusion of the oxygen that way. And because the blood is moving, bringing fresh blood in with high oxygen concentration, the difference in concentration remains high. Okay, so, and also the capillary being one single layer of cells, which means it's very thin, the diffusion is more efficient. So three main adaptation. One is the thickness, which in fact is very thin, the capillary being very thin, made up of one single layer of cells. Blood moving maintains the high concentration difference, and the third one being the sheer number of the capillaries. There are so many of these in each tissue. Going into each tissue, it branches into very thin capillaries, which means high surface area. So three main adaptations. This diagram here shows only the diffusion of carbon dioxide. As you can see, blood again moves in the same direction as this is the same diagram. Blood moves in that direction, but carbon dioxide moves from the cells to the blood flow, and that's because there's a higher concentration of carbon dioxide here in the cells, because you have respiration taking place. Carbon dioxide is produced as one of the products, so that has to be removed constantly, and that diffuses into the bloodstream, which moves to the lungs where it's breathed out. Okay, so that's what that describes the diffusion as it happens in the tissues. In plants, leaves are special organs which have special adaptations for efficient transfer of gases, exchange of gases. As you probably aware of this, oxygen is made in the leaves during photosynthesis, which has to leave the leaves. So they leave through these holes here, which is called stomata. They are on the underside of the leaf. This is the top layer of the leaf, and you have the other layers of tissues, special tissues here. You can see the tissues that bring in the water, the xylem tissues here on the top, the phloem, which is here, which takes the glucose that's made in the leaf during photosynthesis. Okay, but we are now focused on how the gases come in and out, how the gas exchange is efficiently happening in a leaf. So let's set our focus on that. So you have these holes which allows the gases to come in, which are called stomata. So stomata, the sheer number of them itself is an adaptation of a leaf for efficient gas exchange. The second adaptation is this loosely packed layer of cells, this tissue, which is called the spongy mesophyll, and that gives a large surface area. Okay, so that's the name of that tissue. That means because it's loosely packed, you can see the air can just freely, easily flow around them, and that's why they're called spongy mesophyll, because they're not closely packed like this. So that improves the surface area, that increases surface area. So very large surface area is one of the features or one of the adaptations of a leaf, particularly in this layer. So you need to mention this layer. Large surface area in the spongy mesophyll layer makes high surface area possible for the gases to diffuse in and out, and then the stomata. So these two adaptations are really important when you consider a leaf. Let's consider other natural factors such as a windy day. So what happens to the transpiration, the rate of transpiration on a windy day? So for instance, let's assume that water is diffusing from inside the leaf through the stomata outside, and the wind is blowing that way. So wind carries away the water, or it may be oxygen or anything that's coming out of the leaf and diffusing through the stomata outside. And it's carried away by the wind. What happens is fresh wind arrives in this area, which has got a low concentration of these things. For example, a low concentration of oxygen here, or water in this area, compared to inside of the leaf. So there is a bigger difference in the concentration of substance from comparing inside of the leaf to outside of the leaf, which means higher, the faster will be the rate of diffusion. As you know, the higher the difference in concentration, the faster will be the diffusion. So other factors such as wind, blowing on a windy day, can increase the rate of diffusion. Is something to bear in mind. If you consider diffusion of water, for example, how fast a leaf or a plant loses water through its leaves on a sunny, bright sunny day. If it is a dry day, what happens? So dry day, normally outside is got low concentration of water, just because it's a dry day, low moisture in the air. So over here, low concentration of water compared to inside the leaf over here. So high concentration of water, compared to outside the leaf, which means the difference in concentration is high, and this happens on a dry day. So you need to consider factors such as dry day, or which is the other way of describing that is humidity. On a low humidity, low humid day, less humid day, you have a higher rate of diffusion. On a windy day, you have a higher rate of diffusion because substances are carried away by the air, maintaining the high difference in concentration between inside the leaf and outside the leaf. In a plant, you have roots which branch into root hairs. So let's have a look at how this is an efficient adaptation to absorb or to take in water and minerals from the soil. So these brown things, they represent soil particles, and the blue part represents water with dissolved minerals in it. And these root hairs contain millions of these type of cells called root hair cells. Okay, now just because of the sheer number, it's got a very high surface area, because when things are broken down into smaller things, you have very high surface area. So the root is already adapted for efficient diffusion. So when you have high surface area, more water comes into contact with the root. In fact, more water comes into contact with the millions of root hair cells which are everywhere on these root hairs. Okay, so that is how plant roots are adapted for efficient diffusion of water as well as minerals that are taken into the plants. Here is a question to check your progress and understanding. What you see here is a model of a cell. As you can see, the dotted line represents that the cell membrane is permeable, semi-permeable to be precise, as it only allows smaller molecules to diffuse through, and it does not allow the bigger molecule to diffuse through. Now this cell contains the bigger molecules here, as you can see in orange. So those are bigger molecules, and the red dots represent the smaller molecules. The cell is placed in water. Now the question is, after a few hours, what do you think will happen to the number of the big molecules, the number of the smaller molecules? So have a think about this, maybe draw a diagram and then check the answer when you're ready. So after 2 or 3 hours, this is what you would observe, or one would observe. The smaller molecules have diffused out. As you can see, the smaller molecules are evenly distributed inside of the cell compared to outside. You got pretty much the same number. However, the bigger molecules are unable to come through the pores as they are too small for their sizes. The bigger molecules, just being because of their size, are not able to diffuse out. So the only diffusion you see is the smaller molecules. What you see, this is a typical case of say glucose molecule compared with starch. So if you have glucose made in a plant cell, cell wall is not shown for simplicity. So if this is smaller molecules are glucose and the bigger molecules are starch, then you can see the glucose being able to diffuse out of the cell membrane and starch not being able to. But this is true for many smaller molecules which are easier to diffuse through cell membrane, such as you have oxygen, carbon dioxide. They're able to freely move in and out of cell membrane, whereas bigger molecules such as starch and protein are not able to move through cell membrane out through the pores. What you see here is an idea known as osmosis, but this is not in our B2 syllabus, but sometimes questions are based on this idea. Although you don't need to know the term osmosis, but you need to be able to say in terms of diffusion what is happening. So it is really important, particularly for students who are sitting for higher level higher tier exams. So what's happening here is you have a semi-permeable membrane here. So what you have is that is a semi-permeable membrane, which means it allows smaller molecules to pass through. Okay, and you have larger molecules such as starch over here. So we can say that is starch, or even sugar is too big to pass through. Remember, glucose can pass through because that being a smaller molecule, but sugar is not able to pass through because it's slightly bigger. Let me change this to molecules. Okay. Osmosis is in fact a special case of diffusion. If you observe this closely, what's happening here is sugar stays where it is. It's not able to pass through, but the water moves this way. So after a few hours, what you have is the water level rising here. Over here you see the water level rising, and that is because there is higher water concentration on this side compared to here. Now, as most part of the substance, the liquid taken by sugar molecules, and the sugar molecules also has a capacity of retaining some water molecules around it. So the water concentration is higher on this side, and it moves by diffusion or osmosis in this case into where the water concentration is lower. So it sounds very much like diffusion, but this is the case of water molecules moving. That's the only difference. So, and it is because of a solute. So here you got a solute in the form of sugar, is our solute, and because of that, what you have is a lower water concentration. Here you got a higher water concentration. So what you have here is the water moving from a higher concentration of water on this side of the semi-permeable membrane towards the where the water concentration is lower. So lower concentration of water on the right hand side. Let's apply the idea that you've learned in the under osmosis so far in a very common experiment that we do in schools, where you have potato cylinders cut from potato, which is here. So these are cylindrical potato chunks cut off from potato. As you know, potato cells contain sugar. Okay, so depending on the sugar concentration, water either moves in by osmosis or moves out of these potato cells by osmosis. Okay, now carefully consider where you have a higher concentration of water and where you have lower concentration of water, because applying that principle you can predict which of these potato will increase in mass, okay, by the movement of water, in other words by osmosis. Okay, so if you think about this, the first one, let's have a look at the first diagram over here. You have a higher concentration of water over here. I'm going to say H to represent higher concentration of water, as I've said here, L to represent lower concentration of water. So inside the potato cells you have lower concentration of water. How am I able to say that? It's because we know all potato cells contain roughly on an average that much of sugar, so 0.35 mole per dm cube solution. So that's the concentration of sugar inside the cells of potato. So obviously, this being distilled water, you have a higher water concentration compared to the water inside the potato cells. And potato cells have semi-permeable membrane, which allows water and other smaller molecules diffuse in and out. So you have millions of these semi-permeable membrane, and inside those cells you have a lower water concentration. Now it's easy to predict what will happen. Water has to move, or will move, from an area of higher concentration to an area of lower concentration. So it will move inside the cells. So the effect of that is, after a while, say an hour or two, you will, if you weigh the mass of the potato cylinder that you cut, it will weigh higher than at the start. So before you put it in distilled water, you weigh it, record its mass, and after you put it in distilled water and leave it for 2 hours and weigh it again, the mass will increase. And you should be able to explain why. Because the water has actually moved from an area of higher concentration to a lower concentration by osmosis. Let's have a look at this solution here. Now this is 2M sugar solution, which is higher than this concentration. So you have a higher water concentration here. Okay, higher water concentration, lower water concentration. I hope you're not confused with this. You have a concentrated sugar solution here, which means if you have higher sugar concentration here, you will have a lower water concentration. So just bear that in mind. Stronger solutions have less water. Okay, so this being stronger solution, you have a lower water concentration, hence I've written L here. This is a dilute solution, a weaker solution of sugar, which means it's got more water in it, so higher water concentration. I hope that's clear. So now, as we can straight away apply the osmosis rule, it goes from an area of higher concentration to an area of lower concentration of water. So it's really important that you say it's a concentration of water when you explain what's happening here. Okay, so as you can, it's easy to predict now what will happen. Now, if you leave this setup for a while, for say 2 to 3 hours, this potato chunk will lose its mass. Okay, so if you weigh this before this experiment and after, you will find the mass of the potato cylinder reducing. Let's have a look here. You have 2m sugar solution here. So compare with that one, you have a weaker solution, which means you have a higher water concentration here. Less sugar means more water. Okay, so here you have potato cylinder with 0.35 m, so you have lower water concentration there. So high sugar, low water. So now it's again easier to predict what will happen in this case. The water will move from an area of higher concentration to a lower concentration. So water will move in, the mass of this potato cylinder will increase. But it will increase more here compared to here, because the difference in concentration is higher here compared with this one. There is a small difference in concentration of water here, whereas here there is a large difference in concentration. You have 2m sugar solution here compared with 0.35, as you can see. So there will be a greater loss of mass here, a smaller increase in mass. So smaller increase in mass, higher increase in mass, bigger decrease in mass, a loss of mass here. So just to summarize, water moves in by osmosis because it goes from an area of higher concentration to an area of lower concentration in this case. In this case, higher concentration to lower concentration of water. Remember, you have to mention water. So that means this potato chunk will lose mass. Here, higher concentration to lower concentration of water. So this potato cylinder will increase in its mass. Here is the result of the experiment that has been described so far. What you have here is the concentration increasing along here, this axis, concentration of sugar solution in the beaker, and what this shows is the change in mass of the potato cylinder. Now you can see the potato cylinder has increased in mass when it is in a dilute solution, which means when it's placed in a higher water concentration. Okay, so as you can see, the water has moved into the potato cylinder by osmosis, and it's resulted in an increase in mass. Slightly less increase in mass, but nevertheless there's an increase in mass, a smaller increase in mass. Here there is no increase in mass. What that shows is that point is known as the isotonic point. It's just out of interest. It is not in an AQA syllabus, but it's just worthwhile knowing. But you won't be asked or used or expected to use that term. But what that means is at this point here, you have the sugar concentration in the potato cells exactly equal to the sugar concentration in the solution in the beaker. So where it crosses the x-axis, it shows there is no change in mass, which indicates osmosis. There is no net movement of water. Although water is free to move across the membranes of the potato cells freely, what's happened here is there is no net movement. So the result is the potato cylinder has not changed in mass. It remains as it is. So the mass has not changed. Here there is a decrease in the mass of the potato cylinder, showing that water has moved out of the potato cells because that is being placed in a stronger sugar solution. So as you can expect, there is more water concentration in the cells of the potato that has moved out into the solution of the beaker. So hence there is a decrease in mass. The bigger the difference in concentration, the higher is the rate of osmosis, and you can see that it reaches the limit of osmosis. Okay, so all this shows is if there is a difference in concentration of water, the water does diffuse and move out of through a semi-permeable membrane by osmosis. Okay, just to summarize that, there's an increase in mass, no change in mass at this point here, and there is a decrease in mass here. You should now be able to describe adaptations of animals and plant organs that make the rate of diffusion faster. So for example, leaf, lungs and special organs, you should be able to describe the adaptations to increase the rate of diffusion. You should also be able to explain the importance of diffusion in living things. So why is it important? It allows you to take the oxygen in at a faster rate, and how it is important for living things to survive. For example, removing carbon dioxide as a waste product, and it only moves out of the lungs when it's breathed out by diffusion from the blood into the lungs. You should also be able to explain how the rate of diffusion is increased by special adaptations of animal organ and plant organs. This is explaining, here it's describing. You should also be able to explain how water moves through semi-permeable membrane by diffusion. That brings us to the end of this video lesson. I hope it has been useful to you. I know this lesson's been pretty hard. It's meant to be for those who sit for higher tier exams. It is hard. Try to understand this by doing some extra research. So best wishes and see you in my next video. Bye.